Nucleic acids are polymer of nucleotides. Examples of nucleic acids are DNA and RNA. A nucleotide is made up of three components: a 5-carbon pentose sugar, a nitrogenous base, and a phosphate group. The base is attached to carbon atom 1 and the phosphate group is attached to carbon atom 5 of the pentose sugar. Ribose is a pentose sugar with a hydroxyl group attached to its carbon atom 2, whereas deoxyribose has a hydrogen atom attached to carbon atom 2. A nitrogenous base is a base with a ring structure containing nitrogen. In DNA, there are 4 different bases: adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, and the other two, cytosine and thymine, are pyrimidines. RNA contains the 4 bases adenine, guanine, cytosine, and uracil in place of thymine. Phosphoric acid is joined by condensation to the pentose sugar. It gives nucleic acid their acidic property. Nucleotides are joined by phosphodiester bond through condensation process to form a polynucleotide strand. A phosphate group is linked between the carbon atom 3 of one pentose sugar and carbon atom 5 of the next pentose sugar molecule. In 1953, James Watson and Francis Crick deduced the double helix model of DNA structure. Watson and Crick proposed that DNA is a polymer consisting of 2 polynucleotide strands. The 2 polynucleotide strands are coiled in a right-handed spiral to form a double helix. The surface of the helix shows alternating major and minor groove. The 2 strands are antiparallel. One strand runs 5' to 3', while the other strand runs 3' to 5'. The 2 polynucleotide strands are held together by hydrogen bonding between bases in opposite strands. The base pairing is precise. Adenine is linked to thymine by 2 hydrogen bonds, and cytosine is joined to guanine by 3 hydrogen bonds. The base pair are stacked 0.34 nanometer apart. The double helix makes one complete turn, that is 360 degrees, for every 10 bases pairs of nucleotide, or 3.4 nanometer. The diameter of the helix is 2.0 nanometer. RNA consists of a single polynucleotide strand. There are 3 main types of RNA: mRNA, rRNA, and tRNA. Messenger RNA molecule is made up of single-strand polynucleotide. It carries the genetic code transcribed from DNA in the nucleus to the ribosome in the cytoplasm where protein is synthesized. Ribosomal RNA and protein are the structural components of ribosome. Transfer RNA are the smallest RNA molecules containing 75 to 80 nucleotides. There are different tRNA to carry different amino acids to the ribosome in the cytoplasm. Differences of DNA and RNA. Nitrogenous base of DNA nucleotide can be adenine, guanine, cytosine, or thymine, whereas nitrogenous base of RNA nucleotide can be adenine, guanine, cytosine, or uracil. The pentose sugar of DNA nucleotide is deoxyribose, whereas the pentose sugar of RNA nucleotide is ribose. DNA consists of two long polynucleotide strands, whereas RNA consists of a shorter single polynucleotide strands. The two polynucleotide strands of DNA coil around each other to form a double helix, whereas in RNA, no double helix is formed. There are only one type of DNA, whereas there are three main types of RNA: mRNA, tRNA, and rRNA. DNA carries genetic information, whereas the three different types of RNA are important in protein synthesis. DNA is manufactured in the nucleus and mainly found in the nucleus with small amount in the mitochondria and chloroplast, whereas RNA is manufactured in the nucleus but found throughout the cell. DNA is chemically very stable, whereas RNA is chemically less stable. DNA has larger molecular mass, whereas RNA has smaller molecular mass. DNA is susceptible to UV damage, whereas RNA is relatively resistant to UV damage.
Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder, it has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have three parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree. That helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, or abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles. But we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so that you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also, how many base pairs? The answer: each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATTGAC, can you complete what the complementary DNA bases would be for the other DNA strand? The answer: so remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases? The answer: so remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.